Good morning, everyone, and thank you for joining our grand rounds today. We are very happy to have Dr. David Auerbach, who is an assistant professor of pharmacology and medicine at SUNY Upstate Medical University. He is involved with and has chaired numerous epilepsy and SUDEP patient advocacy and scientific groups. Through interactions with the families of patients with severe genetic diseases, he developed the passion and determination to advance the understanding of electrical diseases of both the brain, and specifically seizures, and the heart, arrhythmias. His research program includes scientific approaches that span from molecular, cellular, animal, and clinical research approaches. He made important insights into the prevalence, risk factors, and mechanisms for seizures and cardiac arrhythmias. In genetic models of epilepsy, he showed it is critical to look outside the brain as these mutations lead to alterations in cardiac electrical function. Following a bench-to-bedside paradigm, he reported cases of cardiac EKG abnormality and near-lethal arrhythmias in patients with severe genetic forms of epilepsy. Applying his clinical database analytical skills, he demonstrated that patients with a classically studied inherited cardiac arrhythmias, disease, long-term QT syndrome, are at an increased risk of seizures. He developed a novel mutant rabbit model of long QT syndrome, which reproduces the neurocardiac pathology seen in patients with this syndrome. It enables him to conduct translational studies to investigate the mechanism for arrhythmias and seizures in long QT syndrome. He is passionate about building and mentoring a multidisciplinary research team, recruiting people from around the world to Upstate, developing tools to improve healthcare for underserved population, and advocating for epilepsy patients who are often socioeconomically disadvantaged. Again, welcome and thank you so much for being with us today.
Thank you very much. Thanks for this opportunity to present. All right, so based on the introduction, you can see that my lab takes a very multi-system and multidisciplinary approach. And some of this, I happened to stumble into it, which I'm going to kind of tell you a story about. But just to give a brief introduction here, as many of you know, people with epilepsy are at a 24-fold increased risk of sudden death. And many of these cases of sudden death are due to SUDEP, sudden, unexpected, non-traumatic, non-drowning death in an individual with epilepsy, whether witnessed or unwitnessed, in which a post-mortem examination does not reveal an anatomical or toxicological cause of death. And this is a big overarching goal of our lab, to really examine the underlying mechanisms for SUDEP here. And importantly here, that people with epilepsy, particularly SUDEP, accounts for 7.5 to 17% of all deaths in epilepsy. And in the general epilepsy population, we're looking at 1 in 1,000 deaths per year, but that number quickly rises to 6.7 or even 14 in some of the drug-resistant or severe genetic forms of epilepsy. And some of the proposed mechanisms for SUDEP include cerebral electrical shutdown or cerebral hypoperfusion, respiratory disturbances, as well as sleep disturbances and failed arousal, particularly that prone position, burying your face, in the pillow, as well as two areas of our lab that we spend a lot of focus is the autonomic dysfunction, as this provides a direct neural connection between the brain and many areas of the body, as well as my screen froze. Cardiac arrhythmias. I'm not sure why it's not advancing. Oh, there we go. Now we jumped ahead. Sorry about that. All right, and cardiac arrhythmias, particularly some of these lethal ventricular tachycardia and ventricular fibrillation. All right, so as I mentioned, we look beyond the classic organ of study. So in genetic ion channel diseases, this may lead to electrical disturbances in the brain in the form of seizures, as well as in the heart in the form of cardiac arrhythmias. But I hope by the end of this presentation today, you'll see that a single genetic ion channel variant can lead to electrical disturbances in both the brain and the heart. And to complement this here, exome sequencing or DNA analysis of 61 SUDEP cases. Through that analysis, they saw that 15% of these variants were associated with genes that are classically linked to cardiac arrhythmias, particularly, which I'm going to be focusing on today, long QT syndrome variants, particularly variants in KCNH2, which lead to loss of function, effects leading to long QT2. So I'm not the only one who's kind of had this idea here. There have been numerous classically studied cardiac diseases as listed here, and there have been, a range from, case reports, case series to even, mechanistic, animal studies that have shown that, variants in these genes not only lead to cardiac arrhythmias, but can also lead to seizures and epilepsy as well. Also looking in the opposite direction, a classically studied neuronal variants, particularly like Dravet syndrome, not only are leading to alterations in electrical function in the brain, but also altered electrical function in the heart as well. And so the first part of my talk, I'm going to talk to you about taking this multi-system, looking beyond the classic organ of study in Dravet syndrome. And the second part of the talk, I'm going to talk to you about long QT syndrome. There. So this is really how the story started. This was during my postdoctoral fellowship in Dr. Isom's laboratory. Luis Lopez Santiago took the brain from a mouse one day. So to get the best use out of that mouse, I said, well, let me have the heart. Let me study the heart of that mouse that day. So I removed the heart, dissociated myocytes. And as you can see here, one of the red group, the mutant group here, had a significant increase in both the transient, that quick peak, and the persistent sodium current. So this increase in depolarizing current, particularly during the action plateau, ultimately led to hyperexcitability in the form of early after depolarizations or ectopic activity, which many of you would see as a PVC on the ECG. So I went back to him and said, what was this mouse here? Because the electrical function in this mouse heart was all messed up. And he said to me, it's really interesting that you say that because this is one of the most severe genetic forms of epilepsy, Dravet syndrome here. And this is due to a variant in SCN1A, heterozygous haploinsufficiency here. And these mice would have seizure onset around 12 days after birth, and many of them would die by 20 days of birth. So I spent the next several years studying these mice, and instead of just isolating myocytes, I isolated the whole heart and did optical imaging of the heart. So what we would do is we'd stain the heart with a voltage-sensitive dye. And with that, you can study impulse propagation. So as you can see here, you have the normal sinus activation in the atria, the LA, left atrium, and then ventricular excitation. And this goes on for a couple beats, which gives me a chance to take a quick drink of water. But you can see here that we have sinus activity here. And then right there, we have some ectopic activity like PVCs, waves traveling in from outside the field of view. And then ultimately, these waves collide and lead to the initiation of this spiral wave reentrant activity there. So for the sake of time, I'm going to kind of skip over this next video. It's basically showing the same thing, but color-coded here. And you can see if I freeze a frame there, you can see that this wave is traveling in a reentrant activity here. And this is what is known to be one of the major mechanisms for the initiation and maintenance of cardiac arrhythmias. These mutant mice also had a significant increase in the duration of the ventricular tachycardia and increase in the frequency. But these are just mice here, and we're wondering, how well does this translate? So we had done some telemetry recordings and we actually captured a PVC, a lethal cardiac arrhythmias, and sudden death. But again, this is in mice here. Does this really translate to humans? So in collaboration with Dr. Parent's laboratory, we got skin biopsies from children with Dravet syndrome, drove them to become myocytes. And as you can see here, just the beating frequency of these monolayers of cells, much faster in the Dravet syndrome ones. And one of them had the largest increase in sodium current, very hyperexcitable. So we got in contact with the family, and this child went to a cardiologist and in fact had some ECG abnormalities such as T wave inversion, as well as flattening of the T wave there. But importantly, this was in a mouse, iPS cells, and an N of 1 case here. So what I've spent the past several years is seeing whether these results translate to humans. So I've been collecting lots of ECG recordings from people with several forms of developmental epileptic encephalopathies. So as you can see here, we've collected over 291 ECGs from people with Dravet syndrome, 406 Lennox-Gastaut, tuberous sclerosis, and then several other non-Dravet drug-resistant epilepsies. And this has been a large collaborative effort with numerous people such as Drs. Donner, Friedman, and Sheffer, as well as we got the baseline screening, pre-drug, before anything, ECGs from the Jazz Pharmaceuticals Epidiolex trial there. So we need a comparator group. So here is a healthy control group. This is from the Pediatric Heart Network. These are all people with normal echocardiograms. And you can see here, all of them pretty much have normal QTC intervals. So I'm plotting on the y-axis your QTC interval, on the x-axis heart rate, and this red dotted line here is the clinical cutoff of about 450 milliseconds. Those of you who say, wait, are these men or women? Most of these were all children, prepubescent, so we're looking about 450 milliseconds there. In contrast, if I take all these DEEs here, or all the drug-resistant epilepsies, you can see we have many above this dotted line. And in summary there's a significant increase in the QT interval in those with drug-resistant epilepsy versus the healthy controls. And all this work and analysis was done by Dr. Justin Ryan, who's a research assistant professor in our group here. So let's tease out who are those people above that dotted line. So first I'll plot here the Dravet group. So the yellow shaded area is above 450 milliseconds, and then above there, that red region is greater than 500 milliseconds. So as I mentioned, 450 is a normal clinical cutoff, but if you talk to a cardiologist, some of them say, I don't get too worried around there, but once you get above 500, that's reason for great concern. So you can see here a significant increase in the QT in the Dravet group, as well as a significant increase in those with Lennox-Gastaut syndrome. And you see many of them are in this yellow region, but only one of them was in that red region. Here's tuberous sclerosis. We have several sitting right around that clinical cutoff there, but none of them really go up very high into the yellow or red region there. So to summarize, based upon those clinical cutoffs, we had a 7.9-fold increase prevalence of clinical QT prolongation in people with Dravet syndrome in particular, compared to the healthy controls. And if you use the 500 millisecond cutoff, you can see, well, none of the healthy ones, we had 7 out of 291, or 2.4% of people with Dravet syndrome having what we call actionable QT prolongation. And as I mentioned earlier, people have different cutoffs. So what I did here, or Justin plotted, was the QTC on the x-axis versus the percent of people above that cutoff. So as you see, as we move to the right here, we have more and more people above that clinical cutoff there. Or basically, I said it the opposite way. We start with everyone above that clinical cutoff, and as you move to the right, you have fewer and fewer. But you can see here that the epilepsy groups, particularly the Dravets in purple here, are right shifted. So if I zoom in on this region right here, you can see that we have many people with Dravet who are above that clinical cutoff, even at these really long QT intervals. So these are not lab mice. We have to account for numerous covariates here. So we performed adjusted regression analysis, adjusted for age, sex, and you can see here that people with Dravet syndrome, LGS as well as TSC, all have a significant increase odds of QT prolongation compared to the healthy controls. And then if we look at based upon clinical cutoffs, again, we see a significant increase here. So at this point, I'm going to transition to looking at this question from the opposite direction. So what I've shown you so far is that in a classically studied neuronal disease, Dravet syndrome in particular, there's cardiac pathologies. But what happens if we look at in the opposite direction? So instead of starting at the mouse level, I started at the human level, and I looked in long QT syndrome. So to give a quick background on long QT syndrome, these are due to mutations that alter the balance between depolarizing and repolarizing currents. So these may be due to a gain of function in the sodium current or a loss of function in long QT2 due to a reduction in the rapid delayed rectifier potassium current or a reduction or a loss of function in the slow delayed rectifier potassium channel current. These are the major forms of long QT syndrome. If we have any experts in long QT syndrome in the audience here, yes, we're up to 12 or 15 different forms of long QT syndrome, but these three make up about 90% of the long QT syndrome population. And all these are going to prolong the electrical activation recovery process. So it's going to prolong the action potential duration, it's going to prolong the QT interval, and if it becomes so prolonged, it can lead to hyperexcitability in the form of early after depolarizations, or the next wave might bite the tail of the previous wave, leading to initiation of arrhythmias such as torsade de pointe. So I had the great fortune of working at the University of Rochester and having access to the long QT registry. So a long QT registry includes 22,000 people. And what's really great about it is let's say, for example, if I had long QT syndrome, it wouldn't just include me. It would include my wife, my kids, my parents. So you have all the affected and unaffected family members. So you can see here, people who are genotype positive for long QT syndrome, there's a threefold higher prevalence of a history of seizures or epilepsy compared to their own unaffected family members, genotype negative family members. And as I mentioned, there's three major types of long QT syndrome, and people with long QT1 and long QT2, which are both loss of function variants in a potassium channel gene, have a significant increased prevalence of a history of seizures and epilepsy, particularly the long QT2s. So we went and did regression analysis, and you can see even after adjusting for your QT interval, age, sex, even concurrent beta blocker usage, you can see that people with long QT2 are at a significantly increased risk of a history of seizures or epilepsy compared to the long QT negative group. Importantly, I want to highlight the beta blockers here. I've gotten some criticism over the years. Could some of this be misdiagnosis? I will say very clearly, yes, there could be some misdiagnosis in here, but we did a lot of analysis to minimize or to check whether that was a major contributor. And you can see by the beta blockers here, if this was purely misdiagnosis, the beta blockers would also be reducing the risk of seizures there. We also did high sensitivity analysis where they had to have a history of seizures or epilepsy, and they had to be prescribed an anti-seizure medication, and all these results held true. And I wasn't the only one who has shown this. The group at the Mayo Clinic has also shown that people with long QT syndrome have an increased prevalence of seizures, and they even went further and got EEG evaluation from these people, and 15% of the patients exhibited epileptiform discharges. And here's the third study where they did EEG evaluations, and you can see, if we're comparing the controls to particularly like the long QT2s, there's a significantly higher prevalence of abnormal EEG findings and epileptogenic activity in the long QT2s compared to the controls there. So from that there, I said, I've made an important association, but I don't understand the mechanism for this. So as you saw, I started at the bench level, at the animal level with the epilepsy story. So I said, I got to bring these results back to the bench. I got to generate an animal model to look deeper into this here. So before digging into there, let me review a little bit about, what is casein H2, and how this channel is formed. So casein H2 encodes Kv11.1 protein, and four of these come together to form a tetramer, and thus forms the rapid delayed rectifier potassium channel current. So as expected, rather, KCH2 is highly expressed in the heart, but it's also expressed in the brain. And it's in the heart, it's critical for myocyte repolarization. So as I mentioned on earlier slides, in LongT2, there's prolongation of the action potential duration or prolongation of the QT interval. But if you pharmacologically block IKR in neurons, it also leads to hyperexcitability. So if you compare the black trace there, where you're applying a step current pulse and seeing the burst firing rate, and then do that again when you give a IKR blocker, you see in red, there's hyperexcitability. And the reason for that is IKR is important for the stabilizing the resting membrane potential, the after hyperpolarization, as well as the final phase of repolarization, in neurons there. So I told you I needed to generate an animal model. But before doing that, I needed to make sure I had an animal model that would be clinically translational to humans. So here I'm plotting out or listing out the heart rates and respiratory rates of many different laboratory animals and the cardiac action potential morphology. And you can see here the rabbits, heart rate and respiratory rate best are closest to the humans, and most importantly here, the cardiac action potential. Look how fast the cardiac action potential repolarization is in a mouse. If I just showed you this action potential, you might wonder, did I accidentally dissociate a neuron? It looks like a neuronal action potential. In contrast, you see the human one here where you have this long plateau phase. Importantly, this long plateau phase is when this potassium channel current becomes activated. So if we were studying this in mice, that current is activating out here, but the cell has already repolarized. So the physiological significance of IKR in mice is very limited. So thus, we decided to generate a rabbit model of LongQT2. So how we did that was using CRISPR-Cas9 technology. We introduced a 7 base pair frame shift deletion in the pore domain of KCNH2. And as KCNH2, LongQT2 is often due to heterozygous loss of function. We studied rabbits with one allele mutated and the other one wild type. And the reason why we went after this pore domain here to make the variant is, going back to the LongQT registry work, I looked at the percent of people with LongQT2 that had history of seizures. And you can see the pore variants conferred the highest prevalence and risk of a history of seizures and epilepsy. So Laura Williams was an MD, MS student in our laboratory, and she did a lot of the molecular analysis. So doing RNA-seq, we can see here that in the mutant animals in both the ventricles and in the brainstem, there's a significant reduction in the total KCNH2 expression. Additionally, here you can see that the mutant transcript only makes up about 12 or 6% of the total KCNH2 expression. So thus, this mutant transcript is very low expression there. In collaboration with Dr. Wachakiewicz's laboratory and Dr. Tucker's laboratory for the molecular analysis, we performed Western blotting, and you can see here that there was about a 50% reduction in the total KV11.1 protein in both the brain here and in the heart there. So building on those results, we decided to perform in vivo conscious EEG and ECG recordings. So we have a setup here where we can use a clinical grade EEG and ECG amplifier, and each rabbit gets its own reference here, and we can record from multiple animals at the same time. So you can see in black are EEG recordings and red ECG recordings from conscious restrained rabbits. And in some experiments, we also get respiratory monitoring there. And everything, of course, has video as well. And what we're doing is we're placing 5 EEG electrodes on the head here in each of the 4 quadrants. And then I place the reference at CZ. People often ask, why did I place it there? It's kind of very non-traditional because there's so much ear and neck muscle that when we tried to place it on the ear, on the back of the head there, we just got so much muscle artifact. And then I have 3 ECG electrodes on the chest. So Kyle Wagner, our lab manager, did a lot of ECG analysis. And as you can see, even by eye here, comparing the wild type ECG versus the mutant rabbit ECG, that QT interval is very prolonged. And we quantified that there. So here's a Q to T interval, and it's significantly prolonged in our mutant animals. And if we start looking at each of the repolarization phases here, if I look at the J to T interval here, it's again significantly prolonged, which means it wasn't driven by some Q or S prolongation. And if you look at the J to T peak here, which is where IKR would be quite important, we again see a significant prolongation. And then somewhat to our surprise, we saw shortening of the TP interval. And people often ask, what does that mean there? We're looking deeper into that, and that could suggest that there's some other potassium channel currents that are important in the late phase, particularly in the dispersion of repolarization transmurally, that could be compensating for that IKR. But that's purely a hypothesis at this time. So now let's look at the brain here. So as I said, we were getting EEG and ECG recordings. So here's an EEG recording at baseline. Those oscillatory activity is due to nostril breathing artifact there. We capture sleep spindles in these rabbits. Of course, movement or muscle artifact. And then here's a case of epileptiform discharges that we captured in one of our mutant rabbits here. So you can see starts out quite focal, and then with time, it increases in amplitude, and it's seen in several of the other leads, suggesting that it may be spreading as well. So here's a here's a second example of epileptiform activity here, increase in amplitude, and then we have some theta waves here. This was all analyzed by a great PhD student in our lab, Veronica Singh. Here's a case of muscle artifact here with rhythmic EEG discharges. So we quantified all this here, and you can see that there was a significant increase in the prevalence of epileptiform activity and seizures in the mutant animals compared to the wild type animals. And if we look at like a Kaplan-Meier type curve, the age of these, here you can see that most of the cases, or not most of them, all the cases of epileptiform activity and seizures are arising in the first 2 months of life. Not only are these mutant animals having an increased prevalence of epileptiform activity, but we also saw a significant increased prevalence of sudden death. And coincidentally, the numbers are the same, 1 out of 68 wild type and 7 out of 37 mutants. It's not the same 7 that had epileptiform activity, but there is some overlap in the ones that had epileptiform activity and seizures. But here you can see that we see sudden death arising across a much larger range of ages. Remember on the previous slide, I showed you that all the epileptiform activity happened in the first 2 months of life, while sudden death is occurring across multiple ages. And we think part of this may be due to the cause of the sudden death. So here are the 7 cases of sudden death. You can see, in the first column, the ages. Most of them are females, but we do have a couple males that died. All of these either were witnessed or recorded or were seen within a couple hours of sudden death there. Interestingly, all these rabbits were found in rigor. And if you think of what causes rigor, it's the depletion of your ATP stores. And, particularly for the cases of like it was, we witnessed the sudden death, or it was last seen, a couple hours, before, like there's 2 hours or less than 2 hours, and it was already, in rigor. It likely suggests that there was hypermotor activity before that, leading to this depletion of ATP stores. We did necropsies on all these rabbits. There were no gross abnormalities seen. And in 2 of the rabbits, we did, during baseline recordings, we did capture epileptiform activity. Surprisingly, one was in a wild type, the other was in a mutant animal. And then for some of them, we were able to actually capture the sudden death, whether it was witnessed and recorded or just video recorded. And in consultation with an epileptologist, we were able to determine whether some of these were sudden cardiac death or seizure cluster mediated sudden death. So I'm going to focus in on these 2 cases here because not only did we witness them, we also got EEG and ECG recordings during it there. So I'm going to start with this first case here. And this is again kind of a interesting, happenstance situation where I got a call from the vet and he said, one of your rabbits is dead. So you need to come down and collect the rabbit and collect tissue from it. So I'm in the elevator going down, and then the vet texts me again and he says, well, no, the rabbit's alive. And I kind of was like, well, is it alive or dead? It was kind of a little strange to be getting a message saying now it's alive, now it's dead. So I said, well, I'll hook it up to my EEG and ECG setup, and I'll be able to determine whether it's alive or dead or not. So I'm recording from it, and like popcorn, all of a sudden the rabbit shot up in the air, and all the EEG electrodes flew off of it. And I'm like, what just happened there? And I did this a couple times, and ultimately I smartened up and I taped the electrodes tight on it. And then you can see what happens in this rabbit here, where we're recording from it. And we get all these epileptiform discharges seen there. And then this goes on for a couple seconds. We're getting some discharges here and there. And then I think in the next screen, yep, now we get some spikes. And then we have this lethal convulsive seizure. This rabbit goes into a full generalized tonic-clonic seizure. You see it goes into the tonic phase here. Unfortunately, the reference popped out. So now you get to see my hands trying to put the reference electrode back in. And then we get recordings again. And you can see this large post-ictal generalized EEG suppression and T-wave inversion. This goes on for a couple minutes. No one ever wants to watch a rabbit die here. So I'm going to kind of skip ahead here. But to summarize this here, we had, leading up to the seizure, we had epileptiform discharges, of course, myogenic artifact as well. And then after the seizure, right here, phase 2 here, we have post-ictal generalized EEG suppression. We have T-wave inversion, which is indicative of ischemia. We have bradycardia. This PGS continues. Now we start getting atrial bigeminy, where we're getting almost like couplets of beats. We're getting AV block, and then ultimately complete AV dissociation, and then ultimately the rabbit became asystolic there and died. So now I'm going to tell you the second case. And John Munkhern, who's a medical student who's working with us, really has got a lot of expertise in doing ECG analysis. Looked at the second rabbit that interestingly died when we were doing a punch biopsy of the ear. So we do a punch biopsy to collect a little piece of tissue for genotyping. And we're recording, and a different medical student came in the room and said to me, what's going on with your rabbit? And we're looking at it being like, what are you talking about? Like, we're right around it. Nothing seems wrong with it. And he says, the rabbit has become bradycardic here. So you can see here that during this period, the rabbit became severely bradycardic here. And then we started getting this atrial bigeminy here. We even started to get ST segment elevation on the ECG with reciprocal depression in other leads. We captured some PVCs in the rabbit. And then ultimately things started to get much worse, where we got enlarged and peak T waves, really indicative of conduction abnormalities, ischemia here. And then finally, the rabbit became severely bradycardic, whereas drop in QRS complexes, and then became asystolic. So these were the 2 cases of sudden death that we captured here. But as I said, we had 7 cases of sudden death, and we captured 2 of them, which we were, by roll of the dice, it was pretty impressive that we caught 2 of the cases, but we really would like to capture all the cases of sudden death. So this is how we've been doing the recordings, where we're putting these pin electrodes underneath the skin, kind of like acupuncture to the rabbits. They don't really flinch at all for it, but we can only keep the rabbits in this restrainer for up to 5 hours. We need to really get chronic recordings. So we recently developed a method where we can chronically record from the rabbits in their home housing cage there. So how we do that is, these 3 people in my group, Laura Williams, Kyle Wagner, who I mentioned, and Nick here, each helped develop this technique. So what we do is we make a small incision on the midline there, and then we tunnel the electrodes underneath the skin to each of the 4 quadrants of the chest there, and then tunnel it around on the back and make a small incision on the back of the head, and then place the 5 EEG electrodes. All those wires run to the center of the back right there, and then those wires come out of this port right on the back of the rabbit there. So, and then that connector, which many of you may recognize as the Rhythm Link MRI connector there, connects to this harness coming down to the rabbit. So we're placing these waffle electrodes underneath the skin subdermally, and this allows us to collect long-term recordings, and we get really great signal with this. We recently published this protocol in Jove here, and this is enabling us to really get a comprehensive assessment of EEG and ECG metrics during various physiological states, high, low heart rates, awake and asleep, as well as to capture the true prevalence and incidence of epileptiform discharges, seizures, and arrhythmias, and then captures under different physiological and environmental triggers for these events. Like, why did this rabbit die today and not yesterday? Was it that, we came in the room and startled the animal? In longitude 2, startled, that alarm clock going off, stress is a trigger for arrhythmias. So we can really capture what are these triggers for these events, and ultimately the multi-system cascade of events that lead up to sudden death there. So here's a recording again from our chronic setup here. We're getting really nice EEG and ECG recordings. And the last part of this talk here, I'm going to present to you how we also are able to use these rabbits now for cardiac safety testing. So as you know, particularly like the lamotrigine FDA warning there, there's the chance of adverse effects of anti-seizure medications, in the heart. So going back to the longitude registry, I showed that people with a history of seizures in longitude 2 had a 4-fold increased risk of arrhythmias compared to the longitude patients who did not have a history of seizures. So, my initial hypothesis for that was probably some of these people just are not well medically managed. So I thought, those who are taking anti-seizure medications, particularly the sodium channel blocking anti-seizure medications, may actually be antiarrhythmic because they decrease the depolarizing current and thus restore the depolarizing, repolarizing balance and ultimately serve to be antiarrhythmic. But I was wrong there. So if we look at people with QT prolongation, particularly those with longitude 2, we had a significant increase risk of cardiac events when people are on anti-seizure medications versus off. Let me take a quick step back and let me explain how I actually did this study. I took only people who had a history of taking anti-seizure medication, and I compared their recurrent risk of when they were on an anti-seizure medication versus when they were off it. Not you on anti-seizure medication versus me who's never taken it. So we're really comparing within patient recurrent risk here. And then I thought, well, it can't be the sodium channel blockers. Again, blocking sodium current should decrease the depolarizing current and thus restore that balance. And I split up by sodium channel blockers, GABA modifiers, and then combined ones. And once again, I was wrong again. It was the sodium channel blockers that were increasing the risk of cardiac events. So I said, yeah, it's super interesting, but I really need to tease out this mechanism. So I picked a sodium channel blocker I had the most people in the longitude cohort taking that, and that was phenytoin. And we could see that phenytoin or dilantin, there's a significant increased risk, again, of cardiac events when they were on an anti-seizure medication. So I want to tease out, this mechanism. Why is this here? So I took HEK cells, which they're human embryonic kidney cells. They're kind of like a blank cell, and I expressed casein H2, that's potassium channel current, in there. And Lou, who is a prep up student in our lab here, did these recordings. So here we're doing single cell voltage clamp recordings. So here is the IKR current here. You see how it's very fast to activate, and you have this plateau here. And this is at baseline. And then he gave phenytoin, 80 micromolar, which would correlate to about normal plasma levels upwards on the 20 micromolar, 20 micrograms per milliliter there, which is the upper end of therapeutic levels. And you can see a significant reduction there. And he quantified that there. So here, the black trace is a current on the y-axis versus voltage on the x-axis trace. And as you move to more and more depolarized voltages, we now have the activation of these potassium channel currents at baseline here. But then when he gave phenytoin in red, this curve was significantly reduced. And compared to if we're giving DMSO, DMSO is basically the alcohol that you dissolve the drug in. So to quantify the activation, the tail current density, you can see that there was a significant reduction in the activation and a modest reduction in the tail current density. So, this is showing here that while phenytoin is a sodium channel blocker, it also is dirty. It's having off-target effects there. So now I brought this back to the rabbits, and I did basically like a clinical trial in it there. Took my rabbits, I randomized them. Half of them got drug first. The other half got placebo. They got the drug for 5 days. Then I washed out for a week, no drug. And then those who started on drug then got placebo or vice versa. And I measured the ECG metrics. And to no surprise again, at baseline, the QT interval was significantly longer in the mutant animals versus the wild type animals. And again, I saw the same effect when I gave the placebo. In this case, placebo was just applesauce because that's what I basically put around the dilantin or phenytoin pills to get the rabbits to eat it there. But most importantly, you can see here the blue traces here is pretty flat at each of the different phases here. And so the drug really did not have any effect upon the QT. In contrast, you can see, in the mutant animals, the QT became significantly longer compared to placebo or baseline. And that's ultimately quantified here, the percent change in the QTC. You can see there was little to no change in the wild type animals, whether they're on placebo or on phenytoin. And the mutant animals, when they're on placebo, there was really no change, but there was a significant increase in the QT interval when the mutant animals were on phenytoin. So to kind of summarize this talk here, I've shown you today that ion channelopathies lead to neuro and cardiac electrical disturbances, and variants in cardiac genes are linked to SUDEP. Starting at the bench level, I showed you that in mice with that model Dravet syndrome have cardiac electrical abnormalities that span from increased sodium current, increased myocyte in human, action potential, hyperexcitability, ECG abnormalities, and then ultimately I showed you, in a several hundred person cohort here that it leads to human ECG abnormalities. Then the next part of the talk I showed you where we start at the bedside with the long QT patients who have an increased risk of seizures. And I brought those results back to the bench and examined the underlying mechanisms. So these casein H2 variants lead to a decrease in the mRNA and protein expression, lead to alterations in repolarization on the ECG, and in the brain led to an increased prevalence and incidence of spontaneous epileptiform discharges and seizures. Some of them were purely electrographic, some included clinical manifestations. I didn't have time to show you today, but when we give a proconvulsant drug, there's a decreased freedom from seizures. Basically, these mutant rabbits are more prone to seizures, and also postictal refractoriness is reduced, which means that they're more prone to having a subsequent seizure. Our rabbits have an increased prevalence of spontaneous sudden death, whether it be spontaneous or drug-induced. And then lastly, these rabbits provide a clinically relevant model of longitude 2 for drug efficacy and safety testing, where I showed you that phenytoin in people led to cardiac arrhythmias. So thus, in our rabbits, we were able to look deeper into that, and it led to increase in the QTC and about a 30% reduction in IKR. So to summarize for you today, thank you for your time. I'd like to acknowledge all these people from our lab, as well as our collaborators for the Rabbit project, as well as the clinical database projects that I did not have time to present to you today in detail, but I do want to acknowledge many of these people who helped with the collection of data for the Dravet work, and of course, our funding sources. So thank you for your time.
Thank you so much for this outstanding talk and for sharing all your research. I think we do have a raised hand. Let me see, one moment.
Oh, and of course, I have to note here, if you want CME credit,
Yes.
text this.
Thank you, thank you. I don't see a question in the chat. I do actually have a question or two. I mean, it's more maybe clinical in a sense that with patients with long QT syndrome, and let's say they get diagnosed with cardiology, is there a multidisciplinary cooperation or approach with these patients between cardiology and neurology? And if so, how do you advise them and follow them?
Yeah, no, that's a great point. When I first started presenting the long QT story, a lot of people kind of were surprised by it. And over time, a lot of epileptologists have come up to me and said, actually, I saw, an ECG abnormality, while they're in the epilepsy monitoring unit. And I see more and more multidisciplinary teams now. Particularly, I'm going to use Dr. Donner's group at University of Toronto Sick Kids. She started a brain heart clinic with several pediatric cardiac electrophysiologists now. And I think this becomes really important, particularly in the drug-resistant epilepsies, the genetic ones, because the same gene that's messing up electrical function in the brain, if it's expressed in the heart, particularly like in Dravet syndrome, it's going to mess up electrical function there. Also, for the pharmacological management, I think it's really important, springboarding off the lamotrigine FDA warning here that, really is, thrown a lot of the epileptologists, into confusion. What should we be doing here? Because lamotrigine really, has great efficacy in the geriatric population, but they're also the ones who oftentimes have heart disease there. So, making sure that we're thoroughly medically managing, these patients. And of course, these cardiac arrhythmias provide one of the potential mechanisms for SUDEP. So getting really a comprehensive care of these patients, I think, is so important. I got a call from a grandmother a couple of weeks ago where her granddaughter has long QT syndrome. And no surprise, based on this talk here, she's having both arrhythmias and seizures. And they're oftentimes trying to figure out, which was it this time? And is one leading to the other? Are we having, cardiac events that are leading to cerebral, hyperperfusion and thus leading to a seizure? Or are we having seizures that are leading to autonomic, disturbances and thus triggering an arrhythmia. And I think it's really important to have this long-term monitoring and to have both the cardiologist and the epileptologist reviewing these recordings together.
Thank you. That's very important and interesting, I would say. We have a question actually in the chat box, and this is from Harvey Sarnet and Laura Flores Sarnet. Do you think that cardiac neuric crests that forms the Purkinje conduction system in the heart is related?
I didn't catch all of it. You're asking about the Purkinje fibers and whether they are like neurons in the heart?
So the Purkinje conduction system in the
Yep.
heart is related, yes, to the cardiac neuric crest.
Oh, to the cardiac neural
So let me
crest.
re-say the question. Do you think that the cardiac neuric crest that forms the Purkinje conduction system in the heart is related?
So, I'd say here, yes, because in the Purkinje fibers here, they do have this longer action potential, morphology, and that's where this IKR current is so important. And IKR, this KCH2 is very important in fetal development. Somewhat distant from what I'm presenting here today, but in mouse models where they tried to create LongQT2 mouse models here, they could not reproduce the cardiac pathologies. But interestingly, part of the reason why I think they also struggled a bit was when they went to make homozygous mice, they were embryonically lethal. And they showed that it had problems with neural tube development here. So KCH2 definitely has a large embryonic impact.
Thank you. I do have a question and about like the last few slides about the effect of phenytoin. Did you also study other like, for example, carbamazepine or other medication that could also affect the QT interval?
We haven't yet, and that is actually our plan and ongoing because yes, I'm showing these phenytoin effects, but one of the criticisms of that FDA warning is is it really lamotrigine that's bad, or is it that all sodium channel blockers, you need to exercise caution in giving that to people with heart disease. So we are going to be looking at some of the other sodium channel blockers like carbamazepine, like lamotrigine, but also looking at some of these other newer anti-seizure medications like cenobamate that may have QT shortening effects as well.
Thank you. I do not see questions in the chat box. So thank you so much for this outstanding presentation. Thank you for your time. Absolutely. So the CME code is on the screen as everybody can see it. And so please, you can claim it by texting to the number on the screen. And if you don't have any more questions, I think we can end it.
Great. Thank you. Have a great day, everyone.
Thank you. You too.